we are going to learn about it's the structure of DNA. So the shape of the DNA is called double helix. It looks like this. The DNA double helix, like we have two strands that are twisted together. Then this is the more simplified structure, right? So you can see that there are nucleotides here. This one is a nucleotide, all right? So this one is a phosphate group, this one is the sugar molecule, and then this is the nitrogenous base. You can see that, right? So this is the simplified structure, this one, and then this one is the the literal structure of the double helix. So the DNA has a double helix structure consisting of monomers, or the building blocks called nucleotide, which link to form long chains called polymers, all right? Then the sugar in DNA, it's deoxyribose sugar. I already told you that this sugar is deoxyribose sugar, which is attached to a nitrogenous base. So this is over here, it's a nitrogenous base. All of this over here, they are nitrogenous bases. Then the phosphate and the sugar molecule are attached to one another. So this and then this are attached to one another. Alright, so they are attached to one another by strong bonds to form long chains, right? Then we are going to learn about the nitrogenous bases. So we have four nitrogenous bases. We have adenine, A, thymine, T, we have cytosine, which is C, and then guanine, which is G. So you should always remember that nitrogenous bases are complementary and they always join together in a specific order. So adenine always goes with thymine and guanine always goes with cytosine. So basically you should remember A, T, which means adenine goes with thymine and G, C. Guanine goes with cytosine. Or cytosine goes with guanine. It's one and the same thing. Then one more thing that you should know is how to draw these shapes, because they'll often ask you in the exam to draw the shape of a nucleotide. So so if the nitrogenous base in the nucleotide is adenine, then you draw this. If it's thymine, you draw this. If it's guanine, you draw this. And if it's cytosine, then you draw this. Like here. Okay, so here you can see that adenine is going with thymine, then guanine is going with cytosine, then again you see the order is the same, right? So you should know how to draw all this nitrogenous bases, right? Because they are important. So I think first you should try to draw and then learn them so that it would be easier for you in the exams. So these nitrogenous bases are held together by the weak hydrogen bond. So you should know this because they can ask you this question in the exam. So you should know that the nitrogenous bases are held together by the weak hydrogen bond. And as you can see here, this is the weak hydrogen bond, right? And I think it's also labeled here, as you can see, weak hydrogen bonds, right? So you should know that. We have learned so much about DNA. So now let's learn about what's the role of it. So DNA carries hereditary information, meaning an information from a parent to an offspring in form of genes. So what are genes? So genes are short sections of DNA that codes for a specific trait and determine physical characteristics. So that determines whether you're going to be short or tall. Either your eye color will be blue or brown, then what your hair color would be. So it determines all your physical characteristics, your genes, right? And it also carries your blood grouping, because your blood group is the same as your mother and your father. Then a gene linked to breast cancer. Also, and the behavior of an organism, whether an organism can be tamed or domesticated. Okay, so most of the DNA strands do not code for anything, so they are known as non-coding DNA, and scientists are still searching for importance of non-coding DNA. Okay, so the some strands in DNA, they don't code for anything, and scientists, and which is known as non-coding DNA. Like sometimes they can ask this question, so you can, so yeah, it's it's good to know that it's known as non-coding DNA, right? So the main function of DNA is controls the functioning of cells, regulate the functioning of genes, and passes on hereditary characteristics. So you should know these things, all right? Especially this and this non-coding DNA, and yeah, this is the simple. So you can just read about it. So yeah. Okay, so all right, guys. So based on what we learned today, you can try this practice question. So you can pause the video, try it on your own, and then you can come again to watch your videos to check your answers, right? So number one is label part 1, 2, and 3. Okay, so number one is an adenine. So I know that most of you confused adenine as thymine because they look one and the same. So let's go back and check. Yeah, right? So adenine is like this, thymine is like this. So let me tell you one trick. You can totally ignore it if you don't like it, but mostly people confuse between adenine, thymine, and then guanine and cytosine. So at the end of cytosine here, you can see a C, right? So whenever you see that C, you should know that it's cytosine because cytosine starts with C. And then it's obvious that the other one is guanine. Then for the thymine, I know that this is not a tail, right? This is definitely not a tail. But then you can just think of it as a tail, and then tail start with T, right? Then also thymine starts with T. So whenever you see something like this, you can just think of it as a tail, and that would be what thymine, right? Then it's obvious that the other would be adenine. I know this is totally rubbish. Then you can totally ignore it if you want, but yeah, but then I hope that it helps this thing. So yeah, let's move on, right? So this would be adenine. Then part number 2, this is um this is a sugar molecule, or you can say that it's deoxyribose sugar. Then number 3, all right? So for number 3, they are not asking what this is, they're asking what this middle part is. So that is weak hydrogen right? So that is weak hydrogen. Sorry for that. That is weak hydrogen bond. Let me just write it here. Weak hydrogen bond, which is this one, all right? Then give the number of nucleotides in the diagram. So how do we count nucleotide, right? So this is 1 because we have a phosphate molecule, we have a sugar molecule, and then we have a nitrogenous base. So we have 1, 2, 3. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So we have total of 10 nucleotides, not 5, but 10. So an easier way to count is you can just count the number of sugar molecules or the number of phosphate molecules. So I go with the sugar molecules. So you can see that they are 10. But then if you want to see, this is a full nucleotide. So yeah. So name 2 places in animal where this nucleic acid may be found. So you can find them in the mitochondria, known as mitochondrial DNA, and then you can found in the chloroplast, known as the no, not in the chloroplast, in the mitochondria and in the uh, nucleus, known as the nuclear DNA, right? What is the natural shape of this molecule? So the natural shape of this molecule, I think by now all of us know, it's known as double helix, right? So that's that. Now draw a nucleotide with the base adenine. I told you guys to know the structure of nucleotide because they'll definitely ask in the exam, especially in term 1. Here it's consisting of 4 marks, but I'm sure in the exam it would be 5 to 6 marks maximum. Alright, so how do we draw this? So let me just quickly draw it. Alright, so my drawing is looking not so good because I'm using a drawing pen here, but with yourself guys, you can basically use a pencil and you can draw a nice one. So simply this is a phosphate, this is a sugar molecule, and with adenine, yeah, if it's adenine you draw this, if it is thymine you draw this, if it is cytosine with the C you can draw this, then with guanine you can draw this. Alright guys, so with this we are done with today's lesson. So if you have any questions you can let me know in the comment section below, and you can also let me know one thing, that if my pace was so fast so that I can reduce it in the next video, or was it too slow so that I can improve it. And I hope this videos help you, and best of luck for your exams ahead. Thank you so much. Bye.